Good morning, everyone. Welcome back to my channel, Molecular Mysteries. Hope everyone is doing fine out there. So, without any further delay, let's get into the class.
Come and fly away with me. Come and fly away with me.
Hey, everyone. Welcome back to my class. How are you all? So, hope you're following my channel. Hope my channel is helpful to all. Whoever is watching it, whoever is following it, I'm so thankful for them, for your encouragement. So, please encourage us in this way. I'll keep posting all the important videos for your doubts and all. Okay, so in the last class we have already discussed about the cell division and on the basic terms in cell biology, right? So let's talk with the second important cell division called meiosis. So, what happens in meiosis? And I also want to conclude this class with the differences between the mitosis and meiosis. So, which may be asked in the exam, like so basic questions, not maybe direct questions, but there would be the haploid, diploid numbers and all. Okay. So, what is meiosis and what happens in meiosis? So, meiosis is considered to be the reductional division. Why? Because the diploid number of chromosomes are made into haploid in the first meiosis. So, before I give you a detailed explanation, let me tell you meiosis is again divided into two important stages, meiosis 1 and meiosis 2. Right? So, what is meiosis 1? This meiosis occurs in the reproductive cells or germinal cells of sexually reproducing organisms. The cells in which the meiosis occurs are called meiocytes. The meiosis in the diploid mother gives rise to four haploid daughter cells which are unidentical with the mother cells. Please remember that meiosis which produces haploid number of chromosomes like n number of chromosomes, n is equals to 23, like they will be whatever the progeny that is obtained after the meiosis will not be identical with your mother cell. Okay, so they show the reduction in the chromosomal number. Okay, now let's go in detail about the stages in meiosis. So meiosis is again divided into two stages like meiosis 1 and meiosis 2. So let me talk about the meiosis 1 importantly, because meiosis 2 is nothing but almost it is similar with the mitosis. Okay, so let me now concentrate on the meiosis 1. So meiosis 1 is a heterotypic division. During this stage, the diploid parent nucleus, which is a 2n in number, divides into two daughter having n and n. I told you it's a division, like it's a reductional division where two haploid daughter cells are formed from a diploid mother cell. Okay, this is what you call the meiosis 1. Okay, so this meiosis is again divided into four phases, mainly prophase 1, metaphase 1, anaphase 1 and telophase 1. Okay, let's take it very slowly. So prophase 1 is again divided into, for your understanding, please don't get confused, these are all the stages of prophase 1: leptotene, zygotene, pachytene, diplotene and diakinesis. Out of these, you should be well aware that the pachytene is considered to be the important stage in prophase 1 because crossing over happens in the pachytene stage. So in the last class I have already discussed about the crossing over, right? How the non-sister chromatids would exchange their genetic material, thus forming the gametes, how thus forming the progeny, right? So that is what you call the pachytene stage. Okay, here in this pachytene you will come across the terms called bivalence, tetrads and all. So please remember, I will tell you. Okay, so this is leptotene, zygotene, pachytene, diplotene, diakinesis. These all come under the prophase 1. The next is the metaphase 1. Metaphase is the best stage of studying the chromosomes because they form an equatorial plate in the cell. Anaphase, you know that the centrioles that are present on either side of your cells would come and helps in releasing the spindle fibers for the attachment to the kinetochore part of your chromosome, and hence the pulling happens in anaphase. And the telophase, you know, whatever is happening in the telophase is opposite of the telophase of prophase, like your nuclear membrane again comes back and your nucleolus get organized again and all. So let's take in simple way. And I told you meiosis 2 is again divided into prophase 2, metaphase 2, anaphase 2 and telophase 2. So don't get confused, okay? It's just for your remembrance. Okay, now let's talk about meiosis 1. So I told you prophase is the prolonged stage. It takes around 12 to 24 hours for the completion of your prophase 1. The leptotene, in what happens in leptotene, I'm just giving you a brief idea. Okay, so leptotene, the nucleus enlarges in size, chromosomes appear in parallel and well separated at the end of leptotene. So I told you, when the chromosomes are like, first of all it appears like a chromatin and later they get slowly condensed and they immediately find their homologous pair, so that they will arrange themselves in the parallel direction to your homologous chromosomes, like. So that is what is happening in the late leptotene stage. What happens in zygotene? The homologous paternal and maternal chromosomes attract each other. I told you what are homologous chromosomes. These are called the bivalents. I know I told you no, these homologous chromosomes are not the exact copy, but they just resemble in some genes wise, and but they different have different type of alleles. Okay, these are not the exact copy. So they just attract the the maternal chromosome attacks the paternal chromosome, forming a homologous pair, and that is how it's called as synapses. Okay, and in this stage only there is the initiation of your spindle fibers formation. Okay, and the pachytene, I told you, this is the most significant substage because it is characterized by the division of each chromosome into two chromatids, right? So hope I'm clear with that. Now, what is bivalent? What does bivalent and tetrads mean? See, I told you in the homologous chromosome, one comes from a parent. One comes from the maternal side and one comes from the paternal side. These are called the homologous pair. So these are called the tetrads. Okay, these are called the tetrads because they have four centrum and like four chromatids. So they are called pachytene tetrads. Okay, these are called the bivalent stage. Okay, so chiasmata, I told you, this is where the crossing over happens. It is a point at which the two chromatids, non-sister chromatids, would fuse, forming a X-shaped structure for the exchange of your genetic material. So that is what you call chiasmata. Okay, and this is what chromosome happens. And I told you there are two important enzymes. Let me give you a better so let me, one second. Okay, so think this is your chromosome. After replication, it got its own exact copy, right? At the same way, this is your, for example, this is your maternal side. It's a haploid. Okay, and in the same way, upon replication, it gives the exact copy of your parent cell. So these both have the same genes present, same alleles for that genes also. In the genetics, I will talk about the alleles. Okay, so in the same way here also, the two chromosomes, the replicated chromosome will have the same chromosome of your parents one. So these are not also having the same alleles. So these are the exact copy. So I told you these are the sister chromatids. And I told you this is what happens during the replication when they enter into fertilization. This will get attracted to the homologous pair. Okay, remember this is 23, this is 23, this is haploid, I'm telling you, just remember it's haploid. Okay, now crossing over is where, so this leads to the formation of, so that is where genetic recombination is happening. So you can see it's a crossing over, and this part is called chiasmata. Once the chiasmata moves from the centromere towards the telomere, this crossing over happens, and you will get the exchange happening. The genes present on the paternal chromosome is transferred. Some of the genes are transferred into the paternal, and some, this is how, you will get a homologous pair. And once this happens, the in the further cell division, you know that it enters into the anaphase 1, right? During anaphase, what happens, the centrioles would enter into the action where it starts producing the spindle fibers. And upon the action of your spindle fibers, the one chromatid will be separated, right? So you will end up having two haploid condition at the end of your meiosis 1. So hope I am clear. Like, see, after crossing over happens, right, after the formation of tetrads and after the formation of the bivalents, you will enter into, these are all happening in the prophase 1, remember. In the metaphase 1, they will arrange themselves in the same, like, in the mitosis, like, they will be arranged in a perpendicular position for your easy pulling of your spindle fibers. After the metaphase, you will end up with anaphase, having your sister chromatids, chromatids being pulled over from both the centrioles, like the spindle fibers, and you will just separate those crossed over chromatids, and thus it enters into telophase, where there is a complete formation of your haploid cells. So these haploid cells will be having only one set of chromosomes, okay, only 23 chromosomes, right? So this is how it happens at the end of meiosis. One ends with having two haploid cells, like 2n is giving rise to n and n. This is what happens in meiosis 1. And you know what is meiosis 2? So this almost resembles with mitosis, but DNA duplication doesn't happen. Please remember, there is no duplication of your DNA in meiosis 2. So what happens is, this 2n has already given, sorry. So in mitosis, what happens? The meiosis 2, what happens? The already formed haploid daughter cells would undergo exponential division, like also called as meiosis 2 or mitosis. So thus again number is increasing, and you will get 4 haploid daughter cells at end of meiosis 2. So hope I am clear. See, let me show you the further things. So pachytene, I told you, this crossing over is responsible for the origin of new species and thus leads to evolution. There are two important enzymes I thought of explaining to you, and those are called endonucleases and ligases. The enzyme called endonuclease would cut the part that has to be transported to the non-sister chromatid of a homologous chromosome. The same way, the ligase would help in ligating that opposite part of your non-sister chromatid for the homologous pair. And thus, this complete evolution would start having the recombination. Okay, so hope this is clear. Now, let's go with diplotin diakinses. Just for your understanding, I am just telling it as brief as possible. So, diplotin homologous chromosomes repel with each other due to the weakening of your synaptic forces. They are not separated immediately as still chiasmata is present. Okay. So, in diakinesis, almost the chiasmata would enter into would completely be leaving the chromosome, and hence it's called terminalization. Okay, and the bivalents become very thick and they start migrating to the nucleus periphery. Okay, and nucleus will disappear. I told you metaphase, where it endains themselves, and anaphase is pulling of your the chromosomes towards the opposite poles and respective poles. In the telophase, the chromosome starts elongating, forming the coils. The nuclear membrane and nucleus reappears, and you already know in the mitosis itself, whatever the happening that happens in the prophase, telophase would be in the same direction. Whatever is missing in prophase, telophase again reappears because that cell division has to continue again. Okay, so thus, meiosis 1 ends with the formation of two genetically different haploid cells. Each daughter cell contains only one set of chromosomes consisting of paired sister chromatids. That is clear, right? Now, in the meiosis 2, it is almost very similar to the mitotic cell division. Each of the daughter nucleus divides to form two cells without change in the chromosomal number. This is important, please remember. Okay, so four stages of meiosis. I told you there are prophase, metaphase, anaphase, and telophase. By the end of meiosis 2, you will be having four genetically different haploid cells. Okay, each cell containing only one set of chromosomes. Thus, n producing from 2n. That is how the meiosis ends up. Okay, now what is the significance of meiosis? It helps in the maintenance of a constant chromosomal number for one generation to the next, and due to the crossing over that is happening, the genetic recombinations would be happening and helps in the origin of your new species, and it also helps in the formation of gametes, I told you, and thus it is useful for your sexual reproduction. So now I want to tell you the important differences between your mitosis and meiosis. See, not like kids, like I just don't want to discuss saying that it is having two phases, it is having three phases, it is having only it is having mitosis covers in, will be having only one stage, it is having two stages, they have prophase 1, they have prophase 2, and also prophase 1. No, I want to summarize it as important as possible because you should be well aware of it. Okay, so like, I'm not taking much time, I think. Okay, so mitosis, it occurs in all the somatic cells. I think they are present all over the body, I can say. Okay, and in the meiosis, let's talk about meiosis. Occurs in the reproductive cells of gonads to form gametes. Okay, this is major important division. Please excuse me if I am not good at writing. Okay, see, this here original cell is diploid. Here also the same thing, right? Original cell is diploid. Great. Now, third important change. Here, please remember the nucleus is dividing only once. What does that mean? Only one cell division. for your convenience. There is no change in the ploidy, I told you. 2n is not becoming 4n. 2n is again becoming 2n. But here in meiosis, the nucleus divides twice, like 2n becoming n reduction, and after that this n upon meiosis 2 will form 4 haploids, right? For your easy understanding, I'm just drawing it. Okay, please remember the main point: the daughter cells are genetic duplicates. They are exact copy with parent, right? But that is not same in meiosis, or not genetically same. They are different, I can say. Okay, so one more point I can say: duration is less. These are all small things here. Duration is less and time of duration is more. Okay, this meiosis only happens in diploid organisms, but this happens in both diploid and haploid organisms. Okay, it happens both in diploid organisms, and mitosis happens both in diploid and haploid organisms, but meiosis only happens in diploid organisms. Example, humans. Okay, so hope I'm clear with the basic information regarding the cell divisions. I'm attaching these important topics in the links also. Please go through it, and please let me know, is there anything questions that can be asked. Please comment me in the comment box. Okay, I'm also providing the important YouTube links for you to understand better. Okay, so see you in the next class, and I'll come with the important topic next class regarding the distribution and sex determination of your chromosomal number. Okay, thank you and have a nice day. See you in the next class. Until then, stay positive, stay healthy, and be kind to everyone. Thank you. Bye bye.